so kind introduction. I myself have devoted for the early detection and diagnosis of bipolar disorder for the last 20 years. And recently we are focusing the so-called proteomic approaches for the early diagnosis or early differentiation among mood disorders. So today I'm going to introduce briefly our current project. I know that it is well known in psychosis that before the onset of the vivid psychotic symptoms, there are periods called prodromal phase, especially with some attenuated symptoms. This period is very critical and important as there are so many reports that the brain changes or the other biological changes already started during the high risk or the prodromal phase of the disease. And when we observe the, sorry, I pressed a different one. Okay, good. Oh, this one, okay. About 15% of the high-risk patients are converted to overt psychosis a year. There is a very similar concept of prodromal or early phase of bipolar disorder, but actually it is very hard to define the prodromal or high-risk stage of the bipolar disorder because, as you know, most bipolar patients can experience very long time of depression before the vivid manic symptoms. So as a result, there is very limited approaches for the high risk or prodromal phase of the bipolar disorders. And most studies are focusing the genetic high risk or vulnerability of bipolar disorders. One of the problems of the research of the high risk patient is that, that they just present vague symptoms, insomnia, anxiety, worrisome, doubtness, something like that. And that is not, that directly related to any specific diagnostic disorder. That they can develop into anxiety, they can develop into depression, and sometimes they can develop into schizophrenia. And the problem is, again, that no high-risk patient of schizophrenia do not come to the clinic to us with the labeling that he has a chance to be schizophrenia. And that is the same also for the bipolar disorder and major depression. They just present very vague symptoms at the present. So it would be great that if we have some good marker, biological marker, which can predict that any given specific patient develop into a given specific disorder. So there are many researchers using imaging, cognition, and other markers. However, we know that still we don't have any valid or reliable biomarker which can predict the progress or the onset of a specific disorder. So when considering the other biomarkers, the blood biomarker have some merit because that it's easy to get. It's cheap compared to the other modalities, and also it can present the state of the disease progress. So especially protein, that all the intracellular function are mediated by the proteins. And if you can measure the changes of proteins, then we can predict the progress of the disease. So that is the idea. We are focusing on proteins these days. And the protein research includes the stages, that the first step is the discovery of the biomarker, which also includes the sensitivity quantification and specificity verification, and is followed by validation, which is actually not easy at all. And that we can find some protein which is associated with a specific disorder, but still there is no reason or rationale that such a changes in that protein is the cause of that illness. Okay. And the next step will be the clinical application. The best way to measure protein is immunoassay. However, it needs specific antibody to a given protein. So it also needs some relatively large amount of blood. Compared to immunoassay, mass spectrometry has some merit that it can measure protein with very small amount of blood. So it allows us to measure repeatedly several or 10 or 20 times with a given small amount of blood if we have a progress of the measurement. And another good merit is that it do not need antibody. That it can screen out all the known peptide or proteins. So it is the best way to screen without any hypothesis. So there was some previous investigations that high-risk group of schizophrenia can be differentiated by 15 analytes from 117 hormones, immune parameters, and some other blood analytics with relatively high predictive values. So this is saying that clinical high-risk group, psychosis converted and normal control with this value, and psychosis converted and non-converted group can be also differentiated by just 15 analytes. A similar study was reported in the mood disorders that the 9 parameters, including inflammation and hormones, some metabolism indices, can be calculated into MDD score by the authors, and which can differentiate MDD subject and control subject with very high possibility, and was also replicated by the same authors. There is another report in bipolar disorder that these authors reported that regardless of the episode type, they can differentiate 16 proteins which can control bipolar disorder and normal control patients, and 7 proteins which appear only in depression, and another 6 proteins which can be found only in manic episodes. So from these results, our lab also measured the proteins of the drug-naive major depressive disorder patients, which included 25 MDD and 25 controls. And we found that with 6 proteins, we can differentiate MDD patients and normal controls with relatively high 0.75 possibilities. And next we added protein and other biological parameter called heart rate variability. And we adopted a machine learning method, and we found that with only 5 parameters, about 80% classification accuracy, we can differentiate MDD and control our patients. So based on these experience, we have designed to differentiate major psychiatric disorder, including schizophrenia, bipolar disorder, and MDD patients, and normal control, to differentiate these severe mental disorder patients using proteomic marker panel. So it consists of about 50 subjects. At this time, we have gathered 50, 49, and 44 patients. And the exclusion criteria was any brain disease or any brain stimulation method or drug abuse history patients was excluded. And also we recruited the same number of the control groups. And from the literature review and from our previous experience, we gathered all the known proteins which was reported in schizophrenia, bipolar disorder, and major depression. And the number of the all proteins was 1,300. So among 1,300 proteins, we reviewed the function of each proteins. And with the MRM method, we extracted 300 proteins that is relatively reliably measured with a considerable amount. So we selected 300 proteins. And next, we set up a methodology which can measure 300 proteins simultaneously at once. It takes about 1 hour with a very, very small amount of blood. So there is a merit of these methods because, that we can control, we can deduce the experimental bias. So we measure 300 proteins simultaneously. So this is the flow of what we did. So among the 50 subjects, we just started with 10 subjects. And after the measuring, that we compared between the groups. So this shows the volcano plate, which shows the first 10 subject of bipolar and control, schizo control, MDD control, and bipolar schizo, bipolar MDD, and schizo depression controllers. And we tried the second set, also the same methodologies, third set, and continued the fourth set. We divided the 10 because, that we wanted to confirm whether the same proteins are validly and reliably measured by the iteration approaches. And this is the summary that, as you see here, for the bipolar and control, when we compared these two, only 4 proteins were measured among the total 4 iterations. And the same for just 1, 3, 2, 1, 2. And when we consider the 3 times discovered proteins, the number is about 12, and here maybe 6 or something, and here 18, and about 10 for these disorders. So about 10 proteins, we can say that they are continuously measured. And next, we tried to whether these proteins can differentiate the diagnostic group. And as you see here, the 98 proteins could differentiate the all diagnostic groups completely. And we tested the 20 sample, and also with 68 proteins, they differentiated the all diagnostic group beautifully. But still, 68 proteins are too much. And there are so many missing values according to the diagnostic group. So adopting the machine learning methodologies, that we selected 13 proteins. The way how to we select the 13 is, that it's just by chance, that in each group, the 13 was the best number to predict the differentiation. So for the schizophrenia and normal controls, this is the result. So the sensitivity was 82 and specificity was 86. And for the bipolar patient, it was much more higher. And for the depression, it was just in the middle between schizophrenia and bipolar. With the statistics, that currently we can divide only 2 groups, schizo, bipolar, and schizo MDD, and bipolar control, something like that. Still, we don't have good statistical approaches to differentiate the 4 groups all together. So we are developing the the ways for the analysis with biostatisticians these days. So based on the experience, our next step is that recruit the prodromal cohort, the high-risk group or pluripotent high-risk group, and follow them for 2 years. And we expect about 20 to 30% of the patients will eventually converted to overt mental disorder. Then we can compare whether these patients exhibit the same the data which we get among the diagnostic groups, and we wanted to find out which proteins has a high predictive value of the converting to psychosis or bipolar or depression. So this is the diagnostic criteria. For the psychosis, we adopted criteria for SIPS. And for the bipolar, we adopted the criteria of the BARS. And it is difficult for the prodromal phase of depression, so we attenuated the diagnostic criteria of DSM-4 or DSM-5, diagnostic criteria of MDD. And we also adopted some pluripotent, which do not meet any the proposed criteria of a high-risk group of schizophrenia, bipolar, and major depressive disorders. So this is the list of the scales what we are adopting to assess the patients during their follow-up. And we started this project 1 year ago, and currently we recruited 67 high risk with pluripotent attenuated symptoms, and among them only 3 converted to overt diagnosis, 1 schizophrenia, 1 bipolar, and 1 depression. We are going to recruit more patients until the end of next year, and we'll follow up 2 more years and wanted to observe that what proteins can predict the actual conversion. Actually, the approaches like this have some limitations. Still, that the number of size is so small, and generally we have adopted cross-sectional designs. So, to solve these problems, we adopted some, that better approaches with the longitudinal, relatively large samples. Of course, it's a small scale, and wanted to find out to overcome the limitations and wanted to find out some good predictable proteins early in the future. So, thank you very much for your attention.